Okay, good afternoon. Let's go ahead and start here. So today is the 3rd of June on a Thursday. I'm hoping you've had a productive week, a little hectic, but productive nonetheless, hopefully. So number of emails that I've been sending out having to do with your lab assignments, also your study guide for your lecture exam, number one, and answering other questions as they come along. So please feel free to keep sending the emails out if you have any questions or suggestions or feedback, and I'll do the same from my end. So as of now, I have extended the deadline for turning in your work because a lot of the students were still playing catch up with getting their lab manuals in and trying to get a feel of what those assignments are like and things of that nature. So as I mentioned, it's fine if you guys turn in the work by Sunday. And then I've already went ahead and sent out your lab assignments for Monday. So you already have a head start, at least for the first day of the week next. The study guide has been sent out as well, lecture exam number one. And then I did some extension there as well. So hopefully the test will be activated from 12 afternoon all the way till 9 p.m. on Tuesday, even though you still have 60 minutes from the time that you start the test on Connect. The lecture quizzes are there. Just a reminder, the deadline is by the end of the semester. So if you're sticking to the schedule, please feel free to take those lecture quizzes on Connect as well. Shortly, I will be sending out the guidelines for your case studies, and those would be worth 50 points, as I'd mentioned before. So I think that is everything that we needed to be brought up to speed with. Unless you have specific questions, please feel free to let me know. All right, so here we are on chapter 3. This is what we will be covering today. And as you can see, the title is Energy, Chemical Reactions, and Cellular Respiration. So again, we are looking at the chemical basis of all life, including human life. And what that means is the one theme that we are seeing here as a recurrent fact is the interconversion of mass and energy, right? So Einstein's famous theory of relativity, E equals mc squared, right? Energy equals mass times the speed of light. So let's take some quick notes here. And so that is operational here as well with living systems, right? Einstein's theory of relativity. So that's what living systems do as well. Mass is accumulated. We talked about metabolism, and part of which is anabolism. So anabolic activities is about building up stuff. And then that mass is converted into energy, either physical, mechanical, kinetic, or thermal energy, right? So these interactions, interconversions are taking place at all times. That is what maintains life. And death actually is marked by a cessation of this interconversion of mass and energy, right? So at that time, the mass and the energy dissipate and they become part of the overall ecosystem, the overall food chain. But you as an entity cease to exist at the time of death. But till that time, these processes are taking place. So what is energy? First of all, energy is your capacity to do work. That's the basic definition. What is work then? And of course, if you're preparing for your test, you might be thinking, well, I'm doing a whole lot of work, right? But in purely scientific terms, no work is being done whatsoever because work is defined as when a force acts and some distance is covered. So you need a force that is acting on something and a distance has to be covered, right? So in other words, if you are pushing against a wall, right? And you might be literally breaking your bones and putting all your might into it, but since the wall is not moving any, no work technically is being done, right? So work equals force times distance, right? And so your ability to do work is what energy is all about, okay? And so we need energy in order to do all of these functions, which I mentioned here: power your muscles for movement, pumping blood throughout the system, absorbing nutrients through the GI tract, right? Exchanging gases in your respiratory system, just simply maintaining your cells. in equilibrium, in homeostasis. All of that requires energy, right? Where do you get that energy from? From glucose. Glucose is the simple sugar, a monosaccharide, which is basically manufactured by plants, all plant life, using sunlight during the process of photosynthesis. So that is what powers this whole chain of uh, energy mass interconversion. It's the very basis, and that's why plants are critical to life all across the planet. Um, so once you get glucose into your system, that glucose is burned and metabolized inside of your mitochondria, which are organelles inside of your cells, to release energy in the form of ATP. And if you remember, we talked about that yesterday. ATP, that stands for adenosine triphosphate. It's a high energy molecule, the so-called energy currency of cells, as you can see here. So there we go. Energy is your capacity to do work. Work itself is force and distance, right? There's two types of energy that are mentioned here: potential versus kinetic energy. It's a simple concept. Potential energy is that type of energy that has the potential to do work. But at that given time, it's stationary, it's not moving. For example, if I hold this pen up here in my hand, I'm holding it up against gravity, right? It's not moving yet, but it has the potential to move if I let it go. So this is potential energy. As soon as I let go and it starts to move, that potential energy changed into kinetic energy. So kinetic means movement, energy of movement, right? So these are the two broad categories of types of energies that we have. And they're interconvertible, of course. You can convert potential into kinetic energy and vice versa. A car that is at a stop signal and then starts moving, potential to kinetic energy, then it's again stops at the next signal. So kinetic energy turns into potential energy there. And the same thing is happening all across your cells. So if you look at these pictures here, whether you're aware of it or not, the fact that you're eating and breathing and those nutrients are powering your life cycles, that's happening. So what you see here is your cell membrane. How do you know that's a cell membrane? You can see the phospholipid bilayer, right? You see those phosphates right there in purple, the phosphate heads, which are hydrophilic, so they're facing towards the water, and those hydrophobic fatty tails or lipid tails in the middle. So that's your cell membrane. What is that stuff here? This blue object, which is embedded in the cell membrane, that is a protein channel, right? Protein channels for moving ions in and out of the cell. And in this case, you're looking at sodium ions. Sodium, again, is one of the major cations in the human body, right? It maintains your nerves into equilibrium and maintains the tonicity of the cell. So these sodium ions are, you can see, there's a whole lot of them gathered up outside, right? Not so many of them inside it. So it's sodium is mainly found in your extracellular fluid which is outside of the cells here. So what happens when this channel opens up? See, they all start moving in, right? Why? Because there's a lot of potential energy, pent up energy, if you will, which is all over here. And as soon as this channel opens, the sodium ions start moving into the cell, all right? And this is passive movement, by the way. This is something called passive motion because you did not have to give any extra energy to the sodium ions here. They're already pent up, so many of them waiting to crash the gates, if you will, to get inside. So as soon as the channel opens up, they start moving in. And so potential energy turn into kinetic energy. All right, what you're looking at here is the insides of your mitochondria. And that is where you generate ATP, okay? And that's a very sophisticated and yet a beautifully simple process at the same time that we will talk about shortly here, okay? All right, so what you see happening here is electrons moving from a higher energy level to a lower energy level as we go, right? In other words, you can visualize a ball which is bouncing down the stairs. So at each step, it's releasing some energy, right? It's losing energy. It's going from a higher level to a lower level with gravity. So this is basically what happens inside of your mitochondria. Electrons, which are in a hyperexcited state, they usually move from one electron acceptor to the next. And at each step, they lose out energy, which is captured to form ATP, okay? So in other words, the kinetic energy or the mechanical energy of these bouncing electrons, if you will, is captured as ATP, which is the energy that you use. Here, these are the electron acceptors, you can see them. The electron acceptors, electrons are being passed from one electron acceptor to the next, and energy is being released and captured at each level. All right, so chemical energy, that is the energy of the food that you take in. So carbohydrates, fats, proteins, whatever type of biomolecule you're ingesting, uh, the chemical energy is contained within the chemical bonds inside of them, right? And so these are some examples of major energy giving molecules. Triglycerides, if you remember, that is how you store extra calories. Fat, including fat, right? Fat and uh, carbohydrates as well. Glucose, that is your primary source of energy. And then ATP, that is the molecule that holds energy and powers the life processes. So these are different types of kinetic energy forms: electrical, mechanical, sound, radiant, and heat, right? And all of those are at work in your bodies as humans right now. So you might be thinking, how's it that I have electrical energy? Well, the fact that you're alive and you're thinking, your nerves are firing, muscles are moving, all of that is actually thanks to electrical energy being generated by these sodium and potassium ions moving in and out of your cells uh, throughout your nervous system and muscles, okay? In fact, theoretically, an average human being is generating enough electrical energy at any given point to light up a 60-watt bulb on their heads if somehow we could harness all of that energy, right? So that's a lot of electricity right there. A 60-watt bulb could be lighted up by a single human being's intrinsic electrical energy, okay? Right, uh, so if you watch superheroes like the Flash and stuff, right? They got a lightning strike and gave them superpowers. Well, as humans, we have superpowers of sorts. We are able to generate this electrical energy in terms of thinking and moving and all of that. Mechanical energy again is the energy of movement. So physically going from one place to another, that's mechanical energy. Sound energy, well, our ears, the eardrums respond to vibrations, sound vibrations, right? And we pick those up as sound. Radiant energy is if you stand out in the sun and you feel that heat, the warmth of the sun on your skin, that's radiant energy, electromagnetic waves from the sun, right? And then of course, heat energy. Approximately anywhere from 70 to 90% of the energy that your body generates is actually lost to the environment as heat. That's thermal energy. A major portion of food that you eat is basically being used to just maintain your body temperature at 98.6 degrees Fahrenheit. So being warm-blooded comes with a big price tag, right? We are not like amphibians and reptiles and fish that are cold-blooded animals, right? So their body temperature fluctuates with the surroundings, but we have to maintain mammals and birds maintain their body temperature at a constant level. And that takes a lot of energy from the food that you eat. So here in this picture, you're looking at the electromagnetic spectrum, okay? And something interesting that you see here is if you look at the visible light, the 7 colors of the rainbow, if you will, right? That's the visible spectrum. It's only a minute portion or chunk of the entire electromagnetic spectrum. You see it goes all the way from the least penetrating radio waves here, right? Then comes the infrared light, just slightly more penetrating, but still invisible to the naked eye. Then our colors, the visible spectrum here. Then the ultraviolet radiation, which is again invisible to us. X-rays, which are more penetrating, and the most penetrating of all, gamma rays. All right. So we are surrounded by and bombarded by all these different frequencies of the electromagnetic spectrum that we can't see. But they have profound effects on our daily lives, right? Radio waves, of course. You listen to the radio, AM or FM. Well, those are waves which are being transmitted from the higher atmosphere, right? And the radio sets pick those up. Similarly, infrared light. This is heat, warmth, okay? So the warmth of the sun that you feel on your skin, you cannot see it technically, but it's still infrared spectrum of the light. And I remember back in the days, for example, my father, he used to be a physician. He was practicing back in the late 1960s and early 1970s. So he used to have this cool looking red infrared lamp. And I was always fascinated with that, seeing it sitting in his clinic where he could plug it in and it emitted this bright red glow, almost like a halogen lamp. And I used to ask him, well, what is this contraption for? And he'd tell me, well, this is for providing relief to people who have like muscle spasms or pulled, strained muscles and stuff like that. Something that you would use your, Bengay or those types of creams now for muscle relaxation. So that was a muscle relaxant, right? So, but that was actually a thing. It has fallen out of favor since, right? We do not see infrared lamps in medical practice that often now. So the infrared lamp in my dad's clinic. Also, infrared cameras, they're often used to pick up body heat signatures, right? For example, if you are looking for wildlife, as an example, as a wildlife researcher or something at night, nocturnal wildlife, you cannot obviously see anything in complete darkness. That is where an infrared camera would come in handy, because you can actually pick up the heat signatures from warm-bodied animals, right? From mammals and birds, at least. Even reptiles and amphibians, they also emit and they glow kind of there. So infrared light, okay? Then of course, the longest wavelength and the lowest frequencies for color red. So if you remember Roy G. Biv, right? That is your spectrum of the rainbow, Roy G. Biv. So R here for color red, it has the longest wavelength and the shortest and the lowest frequency, okay? The color violet is the greatest frequency and the shortest wavelength, as you can see here. So then comes ultraviolet light, which is invisible but quite penetrating. So if you stand out in the sun for too long, it's actually the ultraviolet light from the sun that penetrates your skin. And some of it is good because it helps you make vitamin D, but too much exposure and it can cause skin cell mutations, leading to skin cancers. And skin cancers are the one type of malignant tumors that are on the rise more than any other, all right? In the Western Hemisphere, malignant melanoma is the fastest growing cancer, especially for people with fair skin, blonde hair, light colored eyes. They are more at risk for skin cancer if they expose themselves to the sun's harmful ultraviolet radiation. And why more and more malignant melanomas are being diagnosed in modern times? Well, there's at least 3 factors that come to mind as we speak here that might be responsible for this. Why malignant melanomas? Number one, more and more people are exposing themselves to the sun because being tanned is kind of like a fad, right? Back in the 18th century, as an example, having a pale complexion was considered high status, but times have changed. Now getting this healthy tan is in. So increased sun tanning, right? Which puts you more at risk. Number 2, something called ozone layer depletion. So ozone is this gas with the chemical formula O3. So it's a sister gas to oxygen, right? Which forms this layer higher up in the stratosphere. And this ozone layer basically filters out the sun's ultraviolet light. But thanks to some chemical use by humans, chemicals called CFCs, that stands for chlorofluorocarbons, right? Which are found in products like refrigerators and ACs, air conditioners, spray paints, right? Aerosols, spray paints. So these go high up. They have a tendency to rise high up in the atmosphere, react with the ozone layer and break it down. So when the ozone layer breaks down, all of this unfiltered ultraviolet light hits us directly, all right? Number 3, we have better diagnostic facilities now. So we can catch these cancers in greater numbers and earlier on. So at least these 3 factors conspire together to make malignant melanoma one of the fastest growing tumors in the Western Hemisphere. Then come X-rays. We all know what X-rays are used for, right? Of course, for studying hard tissue in humans, bones and teeth, and in other creatures. And then the gamma rays. These are the most penetrating of all and the most dangerous in terms of causing mutations and potential for cancer. Interestingly, they're also capable of burning cancer cells. So you can also use them for radiotherapy, as you can see. All right, so here we're looking at the 2 laws of thermodynamics, and they're pretty simple, pretty self-explanatory. What are they? Thermodynamics. heat and movement. Dynamics is movement, therm is heat. So the first law of thermodynamics states here, energy can neither be created nor destroyed. It can only change form or transfer from one type to another. It's pretty simple, right? So in other words, the energy from the sun in the form of sunlight, electromagnetic radiant energy, strikes the green leaves of the plants, which convert that energy into glucose by the process of photosynthesis. So that sunlight energy got transferred into chemical energy. And then we eat plant foods or other animal food which eat plants, right? So we imbibe or ingest that chemical energy, which becomes chemical energy within our bodies, and then we use that energy to move about so that it gets converted into mechanical energy. We also use some of that energy to maintain heat of our bodies, right? So what we see is energy is not being created or destroyed. It's simply being transferred or converted from one form to another. And that's what this cycle of energy is, or energy cycle is, okay? What is the ultimate source of all of this energy? The sun. And the sun has these spontaneous nuclear fusion reactions going on, right? Helium, hydrogen is being fused together to form helium atoms on the sun, and that it's like nuclear explosions, like detonating a hydrogen bomb. And that's what's happening on the sun, and that is powering life on our planet here. First law of thermodynamics. The second law of thermodynamics states here that when energy is transformed from one form to another, some energy is lost as heat. And this is inevitable. So as I said, the food that we eat, a major portion of that food is used for nothing more than maintaining our core body temperature at 98.6 degrees Fahrenheit, 37 degrees Celsius, okay? Same in your car engine. So all of that good money that you're paying for putting all that gas in your cars, sad to say, but only 30% of that gasoline, the fossil fuel, is used to power the engine. 60 to 70% of that is lost as heat and exhaust from the car, okay? So even with the most efficient engines, there's a significant amount of heat which is lost to the environment. And so that is the second law of thermodynamics. All right, some questions. Both the movement of sodium from an area of higher concentration to an area of lower concentration and the movement of an electron from a higher energy to a lower energy states are example of what? Potential or kinetic, since they're moving, kinetic energy. Muscle contraction is what form of energy? Mechanical energy. Energy can neither be destroyed nor created. However, according to the first and second laws of thermodynamics, what can happen to it? It can transform from one type of energy into another form. And what is always generated? Heat or thermal energy is always generated here. All right, so we talked about metabolism earlier. It's the sum of all chemical reactions taking place in a living system. Chemical reactions, that is, all right, chemical reactions have reactants on the right-hand side, like A plus B here, and products on the reactants on the left-hand side of the equation, like A plus B here, and the products on the right-hand side, which A plus B equals C, all right? So that's a typical chemical reaction that we are looking at here. So in nature, these are some different types of chemical reactions that take place: decomposition, synthesis, and exchange reactions, all right? Some examples. Here is an example of a decomposition reaction, also called a catabolic reaction. Remember, catabolism or catabolic means breaking down, as does decomposition. So what are we decomposing here? What are you looking at in this picture? Sucrose. Sucrose is a disaccharide. Two simple sugars or monosaccharides joined together. And since it's sucrose, what are those two simple sugars that are joined together? Fructose and glucose. They join together to form sucrose, right? So what happened here? Here's sucrose, a disaccharide. You add water to the system. And what did the water do? It split the glucose apart from fructose. What is this reaction called? We talked about it during yesterday's lecture: hydrolysis. Lysing or breaking apart something by adding water to it. So hydrolysis is what you're seeing here. Sucrose broken down into glucose and fructose. This is a decomposition reaction or catabolism. This releases heat. So catabolic or decomposition reactions release heat. They're also called exothermic reactions. Exothermic, exo, out, exit. Thermic, heat. They give off heat. Here you're looking at an exact opposite situation called synthesis reaction. So what happens here? You have two amino acids, one in blue, one in yellow, right? What happened? You let go of a water molecule. A water molecule is lost, and these two amino acids join together to form a dipeptide bond. This is synthesis, the exact opposite, or an anabolic reaction, also called anabolism. So you're putting two things together. Here, and this is, by definition, an endothermic or endergonic reaction. You have to give energy to this reaction to take place. Rather than releasing energy, this type of reaction absorbs energy, okay? Since it's a synthetic or an anabolic reaction. And this is an example of an exchange reaction. So what we have here is a molecule called creatine phosphate, which looks like this, right? Creatine is found in your muscles as is. Creatine is a breakdown product of amino acids that form your muscles. So all of those bodybuilders and people who are looking to bulk up fast, they take these creatine supplements, and you might be thinking, what is creatine? So creatine is an amino acid metabolite, a breakdown product of amino acids. All that creatine does is it has the ability to provide a phosphate to your ADP molecules to generate more ATP. And remember, you need ATP to lift weights just to live. ATP is your energy currency. So what creatine does is it provides these extra phosphates so you can generate more ATP and lift some more weights and take your exercise regimen to the next level, okay? That's what it is. However, excessive amounts of creatine, it's filtered out by your kidneys. So especially if you're dehydrated or not drinking water, along with your exercise routine, then that's putting a lot of stress on your kidneys. So you have to be careful about the amounts of creatine, and then to make sure that you're adequately hydrated so your kidneys don't shut down or something like that, okay? So how is it this an exchange reaction? Well, this is ADP, adenosine diphosphate, because there's two phosphates attached with adenosine here. So this phosphate was exchanged. This phosphate was transferred here to generate ATP now, triphosphate, three phosphates. So this was an exchange that took place here, okay? So we talked about these two types of reactions, exergonic, also called exothermic, and endergonic reactions. What's the difference? Exergonic or exothermic reactions release heat, and they're all breakdown products. They are all decomposition reactions or catabolic reactions. They release energy. They're exergonic. And then endergonic reactions or endothermic reactions, they absorb heat, and they are synthetic or anabolic reactions. They build the stuff, build polymers from little monomers, okay? So here in this graphical form, you can see the difference between exergonic and endergonic reactions, right? So what's happening with the first type here? Here are the two reactants, for example, glucose and oxygen. What happened? Glucose was burned or broken down with in the presence of oxygen to release energy. So what type of a reaction is this? Exergonic or exothermic. Energy was released from glucose, right? So the product has less energy than the reactants, and this energy was released here, as you can see. Now, in this reaction, something quite opposite happens. Amino acids come together and join together to form proteins, but this is an endergonic reaction. You had to give energy to this reaction to form protein from amino acids. So this is an endergonic or endothermic reaction, okay? In other words, when you are working out, this is what you're doing. You're breaking down glucose and releasing energy, and you're losing weight here. Your energy is being released. When you're sitting down and eating meat or protein food and working out and resting rather, that is when you're building muscles. So anabolic reactions, you have to supply energy by eating food to build stuff, to build your muscle. So here we are looking at ATP cycling, and this is the cycle of life, all right? ATP it starts forming in your cells when you were just a single cell little zygote. As soon as dad's sperm fertilized mom's egg and you were generated as a single cell zygote, that's when ATP was active in you. And so ATP sustains you till the time you die, all right? And you're always making ATP even when you're asleep. The only time that you stop making ATP is when you die. And interestingly, the stiffness of death, how dead bodies become stiff, right? Something called rigor mortis is actually a direct result of running out of ATP when you die. Interestingly, rigor mortis, or which literally means rigidity of death. Rigor, rigid, stiff, mortis, death. So rigidity of death is because of lack of ATP. And how's that? Well, ATP is needed to relax your muscles. When your muscles contract, they have ADP there. So they need ATP in order for them to break apart and relax. But since you're not making any ATP when you die, your muscles in a perpetual state of contraction. So rigor mortis is a lack of ATP. All right. So what's happening here? You're building ATP when you eat food, right? In other words, here's the adenosine part, adenine and ribose sugar together. Here are the 3 phosphates. Here's ATP. You start exercising or just living, breathing, whatever. This ATP splits into ADP. That 1 phosphate is released to release energy to supply you with the energy needed to live. And this is an exergonic reaction, of course, because you're releasing energy, and then ATP got converted into ADP, diphosphate, only 2 phosphates here, right? So then you need to regenerate ATP again to live. So you eat food, and the energy of that chemical energy from that food reattaches that lost phosphate to regenerate the ATP, the 3 phosphates here. So this is your life process from ATP broken down into ADP and then replenished into ATP. This cycle will keep going till the day you die. That's what it is in all living systems, right? Okay, so this is the exergonic part from ATP to ADP, and this is the endergonic part from ADP back to ATP. All right, so how fast do you metabolize? That's the question, right? And every person has a different rate of metabolism. We talked about that earlier. Some people just seem to eat whatever they like, all kinds of desserts and whatnot, and never put on an ounce of fat. So they are the lucky ones who got genetically fast metabolizing types of genes. And then other people who might just sniff food and end up putting on a lot of weight. So slow metabolism, right? Metabolism is primarily genetically inherited. So you just have to look at your ancestors and their metabolic rate, and you pretty much reflect that. But there's a lot that you can do to help yourselves too by eating a healthy diet, by being physically active, not sitting down for long periods of time, drinking lots of water, and reducing your stress levels, right? All of those things will perk up your metabolism. So what determines the reaction rate in a system? So to start a chemical reaction in your cells, you need to overcome something called activation energy, right? You need to supply some energy to the system to start a reaction, and that energy is called activation energy, okay? That is something that you need to remember. And another way that you can speed up your metabolism is by adding enzymes or catalysts to this reaction. So what are catalysts or enzymes? Catalysts or enzymes are substances that lower the need for the activation energy. So they make the reactions go by faster, then reduce the amount of activation energy required to start a reaction. So catalysts or enzymes speed up chemical reactions, okay? What are the differences between reactants and products? Reactants are the substances that join together to form a product. For a biochemical reaction that involves simple chemical structures bonded together into a more complex molecule, choose the most accurate term in each paired in this type of chemical reaction. Okay, so synthesis or decomposition reaction. So if you're bonding together, that's synthesis, right? Bonding together is synthesis. Exergonic or endergonic. Bonding is endergonic. You supply heat or energy to the system. Collective term for this type of reaction. Catabolism or anabolism. Anabolism, this is anabolic when you're joining things together. What molecule is formed from glucose oxidation and used as the energy currency for energy requiring processes within the cell? ATP, that's the molecule. Explain what occurs when the equilibrium is disturbed in reversible reactions by changes in reactants and products. Since this is a reversible reaction, it will readjust itself to reach its initial stage, right? Explain the effect a fever would have on chemical reaction rates within the body. So when your body temperature goes higher up, more than 98.6 degrees Fahrenheit, like 99 or something, your chemical reactions speed up. But if it goes too high, too warm, too hot for longer periods of time, the risk is your protein structure of the enzyme becomes denatured, and denatured enzymes don't work. So that's the risk of high fever. It will denature your enzymes. Your metabolism will come to a halt. You can't live at that point. All right, so enzymes, as we mentioned before, are biocatalysts, right? They are catalysts in living systems. And so let's take a look at what happens when you add catalysts to a chemical reactions versus when you do not add any catalysts or enzymes. All right, so here's a graph. You're looking at two types of reactions, one in blue and one in pink. The pink one is an uncatalyzed, no enzyme reaction. The blue one is the same reaction, only this time around, you have added a catalyst or an enzyme. Which one is going by faster? The blue one. The blue one only needs this much activation energy, and then it can go on. The pink one needs to go a whole lot higher, a whole lot more energy is taken before it can accomplish itself, right? So this is the difference between a catalyzed reaction and an uncatalyzed reaction. A reaction that has catalysts or enzymes added to it needs far less activation energy to get going versus an uncatalyzed reaction that needs a whole lot more of activation energy to get going, okay? So that's what you have to remember. Catalysts or enzymes lower the need for activation energy. They make the reactions go by faster. Most enzymes are proteins. We know that they're globular proteins, all right? So here we are looking at how do these enzymes actually work? Okay, so take a look at this picture here. The blue globular protein here is the enzyme. This is the enzyme, the blue stuff. The orange stuff is the substrate, what the enzyme is actually working on, okay? One thing that you see quite clearly here is the shape of the enzyme is perfectly suited to the shape of the substrate. It's almost like a jigsaw puzzle or a lock and key mechanism. You see how the substrate is perfectly shaped to click into place into the enzyme. What does that tell you? Enzymes are specific. So a specific enzyme will only act on a specific substrate and not on any other thing. It's like if I have a key for my car, it's only going to open my car. I cannot open some other model with my car, at least I hope not, right? Unless you're a successful carjacker or something. So the same thing with the enzymes, right? They are specific to the substrate that they act on, okay? Also, all enzymes have end with the suffix ase. All right, they always have ase, ase at the end. So for example, lactase, right? Proteinase or ligase. So if you have an ase there, you automatically know that you're referring to an enzyme. And what does that enzyme act on or break down? What is the substrate? That's the prefix. So proteinase breaks down proteins. Lactase breaks down milk sugar lactose, right? So there you go. That's how you name enzymes. So what you also see here is enzymes have this place on them called the active site, and that is where the substrate clicks into place. It clicks into place in the enzyme, and then it's called an enzyme substrate complex, and that's when the reaction gets going, okay? So enzymes can be found in a number of places within the body. Some of them are found inside of your cells. An example, DNA polymerase. DNA polymerase helps you form new DNA, all right? As you get older, your enzymes don't work as well. So mutations in your DNA become more common and your chances of developing genetic diseases and cancers goes up higher. If you expose your skin a whole lot to your ultraviolet radiation of the sun, more chances that the DNA polymerase would kind of mess up and more chances for skin cancer then. Some are embedded in your plasma membranes like lactase, which is an enzyme embedded in the walls of your small intestine. And that enzyme helps you break down milk sugar lactose into glucose and galactose. People who are lactose intolerant, such as myself, right? They lack lactase. We had lactase when we were kids, drinking milk primarily, but as we grew up, there was not much milk in our diet. So the lactase that gene was switched off. So we didn't make any more lactase. We became lactose intolerant. So when we drink milk, lactose is not broken down into glucose and galactose. We get cramps, diarrhea, indigestion, lots of flatulence, all of those symptoms, okay? And some enzymes are actually secreted from your cells, like pancreatic amylase. This is an enzyme which is secreted. All right, so let's take a look at how these enzymes work and speed up chemical reactions and speed up our metabolism. All right, here's an example. So let's start with lactase. We were just talking about it, okay? So interesting background to this enzyme, lactase. So apparently people from those cultures around the world who come from like pastoral backgrounds, basically cattle raising and lots of dairy farming, they still have lactase persistent even into adulthood. So people, for example, from Scandinavian regions, right? Sweden, Finland, Iceland, Norway, Denmark, those places, right? Um, so Scandinavian populations and some of the East African populations, like the Maasai people of East Africa, right? They are well known for being able to enjoy dairy products without any untoward side effects throughout their lives, because they come from nomadic pastoralist societies that drank a lot, their ancestors drank a lot of milk throughout their lives, all right? So they are lactose tolerant. But they account for only 20% of the world population. The rest of the 80%, especially people from Asia and Africa, right? Asian and African populations, they retained the original form of the gene. And that gene turns off lactase after childhood. So Asian and Africans are generally lactose intolerant, generally lactose intolerant, because they have the original version of the gene that switches off the production of lactase as they hit puberty and onwards, all right? So we were not meant to be milk drinkers throughout our lives. All right, so in any case, how does that enzyme work? Here's the lactase, and you look at the shape. It's specific to fit into the lactose molecule. Look at that, right? And it perfectly clicks into place. Here's lactose milk sugar. It clicks into place in the lactase enzyme. What does the lactase enzyme do here? It kind of changes its shape, right? And it cracks open the bond between glucose and galactose, and there, glucose and galactose are broken apart, and you are lactose tolerant now because you're able to break down lactose. But what happened to the lactase? Nothing. It did its job and it's ready to go back to its and pick up more lactose and keep doing it. So your enzymes are recyclable. Enzymes don't get used up in the chemical reactions, right? They keep working on and on unless they're overwhelmed. If you have too much, a great concentration of too much of lactose flooding the enzymes, then enzymes can become overwhelmed and they're all occupied. It's like all lines are busy, call it a later time. So that can happen to the enzymes with concentration, increasing concentration, all right? Same here. What are we looking at here? Look at this. This enzyme is glycogen synthetase. It tells you what it does. It synthesizes glycogen. What is glycogen? Remember, glycogen is how humans, us, and animals store their glucose in long chain molecules called glycogen in our livers and then in our skeletal muscles. So how do we do that? Using this enzyme called glycogen synthetase. Again, specific shape, which is perfectly matched to the shape of glucose polymers. So what does this enzyme do? Glycogen synthetase, it takes up those glucose molecules and look, makes a long chain out of them. And then what does it do? It detaches and then it recycles. It can go back doing its job with other ones. All right, so for enzymes to act properly, sometimes you need cofactors. What are cofactors? These are helper ions, as you can see, okay? And they could either be organic or inorganic structures, okay? Cofactors. What does that mean? Remember the difference between organic and inorganic? Organic stuff is derived from living things and it has carbon. Inorganic structures are not derived from living sources and they do not have carbon, okay? So inorganic cofactors will actually attach to enzymes to make the reaction go by faster. Some examples of inorganic cofactors are minerals like zinc, iron, selenium, magnesium, calcium, whatever, right? So these are called inorganic cofactors. The organic cofactors, which means they're derived from living things and contain carbon, those are called coenzymes. Organic cofactors are also called coenzymes. And perfect example of them, vitamins. These vitamins are only made in living things, by living things. Vitamins cannot be found in earth, like per se. You need either plants or animals to make vitamins, okay? But minerals, inorganic cofactors like zinc and iron and all that, they are actually found in the soil's crust. So you can literally dig them out of the soil, okay? So look at that, a number of enzymes, examples given here. What do they all share in common? The suffix ase, hydrolase, isomerase, ligase, lyase. And the prefix tells you what do they do. Lyase, they help you lyse, break down stuff. Isomerase, they act on isomers. Hydrolase, they help with hydrolysis, right? So the name tells you, the prefix tells you what exactly that enzyme is working on. So here are some examples. For example, pyruvate dehydrogenase transfers hydrogen from pyruvate. DNA polymerase helps form DNA. Lactase digests lactose. So pretty self-apparent there, okay? All right, so let's take a look at how well enzymes work under certain conditions. So first, concentration of the substrate. And this is what I was talking about earlier, right? If you give controlled amounts of a substance, the enzymes can work just fine. So as you increase the concentration of their substrate, the enzymes work fine. The chemical reactions start increasing and increasing till the point that they reach a point of saturation. So at this saturation point, all enzymes, all lines are busy. So any more concentration that you of substrates that you add to this mixture, it's not going to make the reaction go by any faster. You have reached the plateau here. So it has plateaued at this level. Okay, so this is the maximum point at which all enzymes are saturated. So you can only increase the concentration of substrate to up to a certain point. Same with your temperature. Okay, so let's take a look. Now, this is an enzyme that is working in the human body. So as your temperature increases from 30 degrees Celsius onwards, the enzyme activity picks up and your metabolism goes by just fine. This is your optimum temperature. This is the maximal temperature at which all your enzymes are working at their maximal optimal capacity. And not surprisingly, this is what? It's around 40 degrees Celsius, all right? So it's a little more than 98.7. It's like when you have a light fever, 99 degrees Fahrenheit. That's when your enzymes are working at the optimal level. But what if you have a higher fever than 99 point something, 100, 101, 102 degrees Fahrenheit? Then your enzymes get denatured and they stop working as well. So here you see your metabolic rate is coming down. The reaction is not happening anymore. And this is why high temperatures, high fevers are so dangerous. Especially in children, because they can develop seizures. The brain is very sensitive to temperature fluctuations. So high temperature is quite detrimental to the brain. You can develop seizures, your enzymes denature as well, all right? How about your pH? Your pH also affects enzymes, right? pH of the body. And we talked about how your normal body pH is neutral. It's about 7.35, in between 7.35 to 7.45, right? And there are substances called buffers that maintain this neutral pH in the human body, right? Okay, so anything less than 7 on the pH scale is acidic. Anything greater than 7 on the pH scale is basic. So there are certain enzymes that work best at acidic levels, okay? So think of an organ in your body which is acidic, the stomach, right? And the vagina. Those are two places where you have an acidic pH. So there are certain enzymes like pepsin. Pepsin is an enzyme found in your stomach, which works best at a pH between 1 and 2 because it's active in your stomach, right? And then there are certain other enzymes like enzymes in your pancreatic juices and bile itself. And these enzymes are best activated at an alkaline, at a basic pH, all right? However, the optimum pH for most human enzymes is right around the neutral mark, in between 6 and 8. Not surprisingly, and that's why your body maintains itself at a somewhat neutral pH in between 6 and 8, 7.35 to 7.45. All right, this is the optimal pH. So if you get too acidic, such as when you develop diabetic ketoacidosis, DKA, if you're diabetic, not taking your insulin, or if you have severe sleep apnea or something, then your enzymes get denatured, your metabolism stops, and your health suffers. Or if you become too alkalotic, like if you're throwing up a lot, protracted vomiting, right? Because of cancer or chemotherapy or pregnancy, vomiting or whatever, and that can also cause denaturation of the enzymes. All right, so next up, we are looking at something called inhibitors. All right, so what are inhibitors as compared to enzymes? So inhibitors, as the term suggests, inhibit enzymes, right? That's why they're called what they're called. So two types of them, what are called competitive inhibitors versus non-competitive inhibitors. And we'll take a look here at an example of each. So what competitive inhibitors do, these are substances that mimic the shape of the substrate, right? And so that they swim around and then they kind of squeeze themselves in here onto the active site and bind to it, thereby kicking out the substrate or preventing the substrate from binding to the active site. And that's why they're called um, competitive inhibitors. They compete with the substrate itself to, to occupy the active site here. Then you have what are called non-competitive inhibitors, like this one. And so what these do is they do not go and bind to the active site where the substrate binds. Rather, they bind to a different site somewhere else, which is called the allosteric site. And by extension, these are called allosteric inhibitors. But by binding here, they cause a crumpling up and disfiguring and misshaping of the enzyme to the point where the substrate cannot bind to the enzyme anymore, okay? So since they're not binding to the active site, but elsewhere, and yet resulting in the crumpling of the enzyme, they are called non-competitive or allosteric inhibitors, as you can see. And they're not influenced by the concentration of the substrate because they're not even vying for binding to the active site. They bind elsewhere, as you can see here. All right. So in this picture, you're looking at a typical metabolic pathway, okay? And you're looking at enzymes throughout. And these are embedded enzymes. You can see this is a cell membrane, right? So by now you should be able to pick up on these visual cues. That'll serve you well for your lab exams because it's a very visual exam. You'll be looking at pictures and specimens and whatnot, right? So again, you can see this is a cell membrane. You have the phospholipids, right? Phosphoheads and the lipid tails here. Okay, so 4 different enzymes are found here. Here's the substrate, binds to enzyme number 1, okay? And the first enzyme does its job. Here's the intermediate substrate after processing. It goes to the second enzyme where it's processed. It goes on to the third enzyme here in a different shape, and then to the fourth enzyme here, right? And here's the end product. Now, what the end product does is, see how it swims all the way back to the first enzyme and binds to the allosteric site, thereby acting as an inhibitor and slowing down the reaction. So the more end products that you make, the slower the reaction. Do you know the term for this? When something goes too high, then you bring it down back to the normal limit. Negative feedback, exactly. That's what it is. And it maintains homeostasis, which is constant internal conditions. So you're looking at enzymatic negative feedback in action right here. So enzymes do 2 things primarily. They either phosphorylate or they dephosphorylate, meaning phosphorylation, they add phosphate groups to the reaction and thereby turning on some enzymes and turning off others, depending on what enzymes you're talking about, okay? In dephosphorylation, the exact opposite happens. So these enzymes take away a phosphate, and therefore look at that, phosphatases, A's again. So that's an enzyme that breaks off a phosphate. So, so these are the 2 primary modes of action for enzymes, phosphorylation and dephosphorylation. What is the relationship between enzymes and activation energy? Enzymes lower or bring down the activation energy. What is the active site of an enzyme? Where the substrate binds, and how does it relate to the substrate? It's like a lock and key mechanism. What is the mechanism of enzyme action, including the role of cofactors? So enzymes bind substrates and break them down to speed up the chemical reaction. Cofactors could be organic or inorganic, and they help the enzyme do its job. Organic cofactors are like vitamins, also called coenzymes. How enzymes are generally named? They all carry the suffix "ase", and the prefix tells you what substrate they act on. How do changes in substrate concentration, temperature, pH affect the reaction rates of an enzyme catalyzed chemical reaction? So if you increase the substrate concentration or the temperature or tinker with the pH, the reaction might go faster initially, but after you hit the optimal point, from that point onwards, it's going to taper down. How are enzymes regulated? Through competitive and non-competitive inhibitors. Competitive inhibitors bind at the active site, thereby kicking out the enzyme, the substrate, while the non-competitive inhibitors bind at a different site called the allosteric site and do the same. What is a metabolic pathway? How substrates go from enzyme to enzyme till an end product is made, and then negative feedback takes over. What 2 processes involve phosphate and are commonly used to regulate enzymes? Phosphorylation and dephosphorylation. So here are some examples of drugs that are actually enzyme inhibitors, and they
do their job by enzyme inhibition. And one of the most well-known examples of such a drug is Viagra, the blue pill, right? And quite an interesting story, a backdrop to how Viagra was actually discovered. So back in the early to mid 90s, pharmaceutical companies were on the lookout for a drug which would be more efficient, more effective, and will have lesser side effects than the traditionally used nitroglycerin for chest pain, something called angina, right? So many different candidate drugs were in the works. And so then they came up with this phosphodiesterase type 5 inhibitor. It wasn't called Viagra back then, but that's how it worked. So now again, focus on the name, phosphodiesterase type 5 inhibitor. So in other words, this drug, which is Viagra or sildenafil, actually inhibits the enzyme called phosphodiesterase. So what does the enzyme phosphodiesterase do? It breaks down the phosphodiester bonds between the between nucleotides, all right? In other words, this drug slows down the breakdown uh, of an enzyme that would cause um, your nitric oxide, which again is a vasodilator. Nitric oxide is a substance that causes your blood vessels to dilate, and the same happens in the penis. And so when the blood vessels dilate, more blood flows to the penis, it becomes erect. So what phosphodiesterase does is it breaks down nitric oxide. But by using a phosphodiesterase type 5 inhibitor like Viagra or sildenafil, you are increasing the concentration of nitric oxide. More and more blood vessels and arteries are being dilated in the penis, and you get an erection. So that's the mechanism of action. So in any case, many drugs were in the works, and Pfizer, the pharmaceutical company, basically came up with this drug, which was a phosphodiesterase type 5 inhibitor. Uh, and so what they did was, as is the case in phase one, phase two, and then phase three trials, mostly phase one and phase two trials, you take two groups of people, right? It's like a double-blind, placebo-controlled study. So one group of patients are given the drug, and the other group is given just a placebo, something like a sugar pill or something, which does not have any active ingredients. Nobody knows, including the doctors and the scientists, researchers, as to who got what. It's all entered into a computer. And then these patients are followed for any improvements or detrimental effects or side effects or whatever for a period of time. And then that's how they gather the data as to whether the drug was beneficial and effective or not. So they were given sildenafil. All of these were heart patients, right? So one group was given sildenafil pills. The other group was given a placebo. And so they were followed over 6 months to a year's time. And when they came back, all of this data was collected. And much to the chagrin of the Pfizer people, Viagra did not, or sildenafil did not turn out to be what it was cracked up to be, right? So there were like some side effects, like problems with vision. People started seeing everything with a bluish tinge, and headaches and stuff like that, right? Severe headaches, and it did not seem to have any greater of an activity as compared to nitroglycerin. So that was kind of like a disappointment. But at the same time, the scientists and researchers also noticed something quite interesting. Uh, Many of these middle-aged, overweight, physically inactive gentlemen were reluctant to return the pills. So that kind of set off an alarm somewhere. And they were like, guess what seems to be going on here? So they called in the patients and they asked them, what's this all about, right? Nobody's too keen to give away the pills now that the study is over. And so then basically they found out that the pill might not have helped much with the heart symptoms, but it really amped up their love lives. And their girlfriends and lovers and wives and whoever were quite pleased with whatever was being given to them. Okay. So that was a eureka moment for the Pfizer people. Now they had the perfect drug to market to a rapidly aging, middle-aged people who are overweight and not exercising and not eating healthy and not in the best of shapes, yet want to rekindle their love life, you could market this drug out to them. And so in 1998, it was patented as Viagra, and it became the fastest selling drug in history. Perhaps not surprisingly, looking at our youth obsessed culture and the unrealistic and unscientific expectations that are thrown out there by the popular media, right? Um, Interestingly, so yes, that was the backstory, the backdrop to Viagra. Uh, at the same time, it also had some unintended benefits. For example, some people found out that, and I don't know, this was maybe an experiment done. If you have any pills to spare, okay, they're quite expensive, so I doubt if anyone would. But if you did, then you ground them into a powder and put them in your house plants. Your house plants will stay rigid and moist and vascular and nicely pumped up and not shrivel up for much longer than usual. Okay, actual fact. And why is that? Because the same neurotransmitter, nitric oxide, that dilates the arterioles in the penis is also dilating the xylem and the phloem vessels, not blood vessels, in the plants. Very same mechanism. That also tells us about our shared heritage and ancestry with all other living things, including plants. The same enzymes, the same hormones that act on you are acting on plants. And that's why most of the plants are used for making medicines for humans and animals. So it's all like an intricate web of interconnected life. Same hormones, same biochemistry. Okay. So yeah, you can use it for your plants. Interesting fact, plant use. Okay. Lactose intolerance, we talked about that before. 80% of the world population, especially Asian and African populations, are lactose intolerant because they inherit the original version of the gene that switches off after your early childhood. So you lose your ability to break down lactose. You drink milk or eat ice cream, as I often give into that temptation. And then much to my dismay, I have to spend a major part of my night in the restroom with all of those symptoms that and signs that are mentioned here. Um, right, so that's basically what lactose intolerance is all about. Okay, so here we are at something called cellular respiration, and this is the equation of life. There's actually two equations of life, photosynthesis and cellular respiration, right? So let's call them that. The equations of life. Uh, and so who knows, even with the COVID vaccines that are coming out, maybe we'll find some unintended side effect and they could be marketed as something else, right? Because viruses are always mutating. So what you're gonna do, just come up with vaccines for every single virus and get a billion shots on a regular basis? We'll see how things go there. All right, so cellular respiration is basically simply breaking down glucose in the mitochondria of your cells to release ATP energy. That's what it is at its very basic level. And all living things respire cellular respiration, except for some anaerobic bacteria. Uh, Photosynthesis is the process which is the exact opposite, where plants take carbon dioxide and water from the atmosphere and join those two together to form glucose and release oxygen in the atmosphere as a byproduct using sunlight energy. Okay, so cellular respiration, as we see here, is an exergonic process, which means it releases heat and it releases energy. Okay, and that energy is used to make ATP, and you require oxygen for that. Okay, here you're looking at the overall chemical equation for cellular respiration. So what does that look like? C6H12O6, that's glucose, plus 6O2, which is oxygen inside of your mitochondria, gives you carbon dioxide and water, which are waste products that you breathe out, plus 34 to 38 ATPs per molecule of glucose. That's what cellular respiration is all about, okay? And so where does it happen? All these steps, they happen inside of your cells. The first step, called glycolysis, takes place outside of the mitochondria in your cytoplasm, what is called the cytosol, as you can see here. And the rest of the two steps after that called the Krebs cycle or the citric acid cycle. And uh, electron transport chain. Those two processes take place within the mitochondria. Okay, something to remember. So here, this is a good picture that basically summarizes cellular respiration for you. So what I want you to get out of this is just the broad overall summary and not the individual steps, because there's a number of enzymes, especially here in the citric acid cycle, no less than 10 to 12 enzymes involved. But I want you to get the overall, the big picture here, okay? So glycolysis, the first step takes place outside of the mitochondria in the cytosol, then something called the intermediate stage, and then the citric acid cycle. Now here, things move into the mitochondria, okay? And then the electron transport chain finally, which again is inside of the mitochondria, okay? So let's take a look at what these processes are. So we're gonna start with glycolysis, and we're gonna go to the summary right away. As I mentioned, I would like you guys to take a broad view of this whole process. So first up, glycolysis occurs outside of the mitochondria in the cytosol. It does not require oxygen, important. This is anaerobic, no oxygen required here. And what happens in glycolysis, look at the term, glycolysis. You're breaking down a glucose molecule, which is 6 carbons, C6H12O6, into 2 3-carbon substances called pyruvate. That's what happens, okay? And in the process of splitting glucose, such as you see here in the picture, into 2 pyruvates, you release 2 ATPs. 2 ATPs are formed here out of this, okay? So that's what you have to remember. No oxygen required for glycolysis, splitting glucose into 2 3-carbon pyruvates. It happens outside of the mitochondria in the cytosol, okay? Then in the presence of oxygen, now everything after that needs oxygen. So if insufficient oxygen is there, then pyruvate doesn't go any further. It's simply converted to lactate or lactic acid. But mostly there is oxygen available because we are breathing. And so under those circumstances, pyruvate actually moves on into the mitochondria and then goes on to the next stages, which are these. So the intermediate stage first, right? So what happens in the intermediate stage, and let's see if we can look at the summary, which is quite messed up here, okay? And look at all the steps of the citric acid cycle, a number of enzymes and steps involved here, but we are not going here. Here's the summary. We're not going to focus on all that. So what's the summary of the citric acid cycle? It occurs inside of the mitochondria. You require oxygen for this process. And the pyruvate that came from your glycolysis, which was the first step, it is converted into something called acetyl coenzyme A, as you can see here, okay? The acetyl coenzyme A go on to take part in the citric acid cycle. At the end of the citric acid cycle, you form 1 ATP, 3 NADH, which is an organic substance, stands for nicotinamide adenine dinucleotide hydrogen. These are electron acceptors, electron carriers. Think of them that way. These are also energy-rich compounds, just like ATP. And 1 FADH for flavonamide adenine dinucleotide formed per cycle. Now, since 1 glucose got split into 2, so both of these are multiplied by 2. So you get 2 ATP, 6 NADH, and 2 FADH2 out of 1 molecule of glucose in the citric acid cycle. So let's keep track of the ATPs. You got 2 ATPs from glycolysis, and now you got 2 ATPs out of citric acid cycle. So that gives you 4. Finally, then we move on to the what? To the final stage of cellular respiration, which is called the electron transport chain or the electron transport system. And this again is happening inside of the mitochondria, as you see here, okay? So what happens in this chain? It's quite an interesting process. So I'll simplify it for you. So again, look, this was glycolysis outside of the mitochondria, right? In the cytosol, where 1 molecule of glucose split into 2 pyruvates. You got 2 ATP out of that. The pyruvate in the presence of oxygen went through the intermediate stage inside of the mitochondria, where you generated acetyl coenzyme A. The acetyl coenzyme A took part in the citric acid cycle here, also called the Krebs cycle. Then you got 2 ATP, 3 NADH, and 1 FADH2 out of the citric acid cycle. Then what? Then things move on to the electron transport system or the electron transport chain. What happens here is that Those two substances that were formed during the citric acid cycle, NADH2 and FADH, they donate electrons uh, to this membrane here inside of the mitochondrium. Now, these electrons are being passed from one electron acceptor to the other, like this. You can see it here in the pictures, right? It's like playing a game of hot potato. You know how you pass the object from one person to the other person fast, or a baton race where you pass on a baton to the next runner and they take the baton and they give it on, pass it on to the next person, and so on and so forth. And so that's exactly what's happening here. The electrons are being passed around from one electron acceptor to each. So each time that the electrons, these hyperexcited, energetic electrons, are being passed from one electron acceptor to the other, that releases a lot of energy, okay? So where does all of this energy released by the jumping electrons go? That energy is used to push out protons or hydrogen ions, same thing, from here out here into this space, okay? So again, the energy of these jumping electrons from electron acceptor to electron acceptor, that energy is used to push out hydrogen ions uh, or protons out here from inside to this space, right? So there's a bunch of hydrogen ions all packed here. Now, what do these hydrogen ions want to do? They want to get back inside desperately, right? But there's only one way for them to get back inside. They have to go through this kind of like a turnstile, like a turnstile. You know what a turnstile is? When that revolving door kind of thing, a revolving wheel where one person goes and then the next. So this is kind of like a turnstile molecule called ATP synthase. In other words, this is an enzyme that synthesizes ATP. So the hydrogen ions go one at a time through this ATP synthase molecule, and it rotates, right? Just like a turnstile, with each proton moving in. And with each turn of the ATP synthase, you generate an ATP here, okay? And so by the time that you're done with all of these hydrogen ions moving in, you would have generated about 32 to 34 ATPs from one molecule of glucose here. All right, so what a wonderful process. It's like a whole machinery, like a whole factory going on. Wheels turning and electrons being passed around and all of that happening inside each of your cell. That's like remarkable, unimaginable if you think about it, right? All of these complicated processes, biological processes taking place inside of your cells without our knowledge. So here's a summary of the stages of cellular respiration. Glycolysis releases 2 ATP, 2 NADH, the intermediate stage, then the citric acid cycle, you got 2 more ATPs, 6 NADH, 2 FADH2. Now these would be the electron donors here during the electron transport chain, right? And so then finally, here we go. 2 ATPs came out of glycolysis, 2 ATPs came out of citric acid cycle, right? And then about 32 to 32 came out of your electron transport chain. So when you add all of those up, you get 36 to 38 ATPs out of a single molecule of glucose. That's how much energy you're getting. And right now there's literally millions of glucose molecules getting inside of your cells. Each one is generating about 34 to 38 ATPs. That's a whole lot of ATPs that power your life, that keep you alive. All right, so cyanide poisoning, interesting. Now, cyanide was used as the weapon or the poison of choice widely during the Second World War. So spies, like wartime spies, both from the Allied army and from the Nazis, they used cyanide poisoning quite commonly because this is a very insidious way of poisoning someone. It leaves no traces in the body, but what cyanide does is it basically shuts down your electron transport chain. So it decouples or uncouples the breathing process and the transfer of oxygen and glucose to your cells and the generation of ATP from that glucose. So in other words, you might have all the glucose you need to make ATP. That glucose is just simply not going through the respiratory cycle. It's blocked. Cyanide blocks out the electron carriers in the electron transport system. Therefore, your ATP production shuts down. You die because of exhaustion, no ATP production, all right? And interestingly, cyanide is found in some natural substances as well, like bitter almonds, right? So theoretically speaking, if you were to consume upwards of 200 bitter almonds all in one sitting, you could technically poison yourselves and die of cyanide poisoning, right? Also, apple seeds. Apple seeds also have residual amounts of cyanide. So cyanide poisoning is something that relies on shutting down the electron transport chain and uncoupling the ingestion of glucose and oxygen into your cells and the production of ATPs out of that. All right. Okay, so what happens when you have insufficient oxygen in your system, such as when you're running a marathon and you're breathing at your maximal capacity, no more oxygen can be delivered to your tissues. You've absolutely hit the wall. What happens at that time? At that time, uh, since there's no more oxygen available, glycolysis eventually shuts down, as you can see here, okay? And all of the pyruvate that came out of glycolysis is converted into lactic acid or lactate, okay? This is called anaerobic respiration or fermentation. And all you get out of fermentation is 2 ATPs. Normally, with enough oxygen, you should be getting anywhere from 36 to 38 ATPs per glucose molecule. But when you're out of breath, you only get 2 ATPs because of fermentation. But those 2 ATPs might just be enough for you to hit the finish line or to just catch your breath if you're drowning or something. And that could be a lifesaver, right? In plants, on the other hand, if you switch from aerobic to anaerobic respiration or fermentation, they make alcohol. That's how you make alcohol beverages, by depriving plants of oxygen using some anaerobic bacteria, and then they generate alcohol, which is used in beverages, okay? All right, so in case that you have run out of glucose, which is your primary fuel for the body, your brain almost exclusively feasts on glucose and nothing else, then your body can rely on, it falls back on other biomolecules for energy, like fats and fatty acids, right? And it can get energy from fatty acids by a process called beta oxidation, all right? And acetyl coenzyme A is actually oxidized aerobically here by beta oxidation. What types of people will be doing this? Do you remember? What types of people would be actually beta oxidizing their fats and generating a lot of ketones? People with diabetic ketoacidosis, people who skipped their insulin shots, they would be doing a whole lot of this, the ketone levels, or if you're on a keto diet, that's what keto diet is about. No carbs, or Atkins diet, previously known as. Uh, so no carbs, but lots of proteins and lots of fats. So your body switches from carbs to fats for energy generation, but you pay a price. Your ketone levels go up, you can end up with acidosis. So you are artificially inducing diabetic ketoacidosis by going on a keto diet, right? So something to keep in mind. So there are dangers of severely restricting carbohydrates in your diet, okay? Because again, like I said, it's like volunteering for a diabetic ketoacidosis. Then amino acids, these are the last ones. If all sources of other sources of energy have been utilized completely, then your body falls back on amino acids. They're typically, proteins and amino acids are not energy sources. They're bodybuilding materials for wear and tear, not for energy generation. But if you're starving, that could be the case here. All right, uh, so what happens when you oxidize amino acids is you convert it to urea. And urea is a waste product which is excreted by your kidneys via urine. So write the overall chemical reaction for glucose oxidation. C6H12O6, which is glucose, plus 6O2, which is oxygen, gives you 6CO2, which is carbon dioxide, plus 6H2O, which is water, and 36 to 38 ATPs. What are the 4 stages of cellular respiration for glucose oxidation? So glycolysis, intermediate stage, Krebs/citric acid cycle, and electron transport chain. Where does each occur in the cell? All of those occur in the mitochondria except for glycolysis, which happens out in the cytosol. Describe glycolysis, splitting of a glucose molecule into 2,3 carbon pyruvates. Where it occurs in the cytosol, if the process requires oxygen, yes. Oh, it does not, sorry. It's anaerobic, the net chemical reaction and the net energy. So you get 2 ATPs out of it. What are the 2 general fates of pyruvate? Either in the presence of oxygen, it moves on as acetyl coenzyme A into the intermediate stage, or it is converted into lactic acid under anaerobic conditions in the lack of oxygen. Explain the enzymatic reaction involving pyruvate dehydrogenase in the, okay, so we are going to move across that one. Uh, Summarize the metabolic pathway of the citric acid cycle. So you can look at the cycle, but I do not expect you to remember and memorize each and every stage and all the enzymes involved there. What energy molecules are produced from the chemical breakdown of glucose during each of the three steps of cellular respiration? ATP. What is the importance of NADH and FADH2 in energy transfer? They are the electron donors to be passed around during the electron transport chain to generate energy, to power the formation of ATP. All right, what are the three primary steps that take place in the electron transport system? We know that. Electrons being donated and passed around from electron acceptor to electron acceptor, that energy being used to pump out hydrogen ions or protons in the outer space, and then finally these protons moving into the space back by turning the turnstile called the ATP synthase and generating more ATP. How many ATP molecules are generated through glycolysis? Only two. How many net ATP molecules are formed through the combined processes that occur in the cytosol and those that occur within the mitochondria? 36 to 38 ATPs. Pyruvate is converted to what molecule if there's insufficient oxygen? Lactic acid. Why is oxygen required to burn fatty acids? Because oxygen oxidizes fatty acids. Beta oxidation is what it's called. And it also generates ketones on the side. Okay, so all done with chapter 3. So hopefully you would have enough time to go over the study guide and use it to the best of your advantage. Also, listening to the lectures in their entirety and focusing on the notes that I'm sending out. Keep working on your lab work that I sent out. Hopefully you'll be getting in your lab manuals and all of that too in the meantime. Keep up with the lecture quizzes on the Connect website and case studies guidelines. I will be sending those out too. I will be grading your lab quizzes over the weekend. And any questions, feedback, and suggestions and concerns that you might have, feel free to let me know. And so have a wonderful weekend, long weekend for us. And take care. And I will see you all virtually on Monday then. All right. Have a great one. Bye-bye.